Can you hear me?
I can hear you perfectly.
Okay, wonderful.
Super. So I'll quickly introduce what we're doing. So my name's John. I'm doing this on behalf of a company called Adivo Associates, who are an independent market research agency. Everything you say today will be reported confidentially and anonymously, and I will be recording the call. But I would ask you to please just avoid saying your name or anything that might identify you during the call.
That's fine.
Okay. So, firstly, just wanted to ask you, just looking at your screen info, you prescribe quite a few different products for haemophilia A, but you mainly prescribe Adenovate, Electate, Eloctate, and Novoate. Tell me, what do you remember or know about those products just off the top of your head?
I miss your last sentence. I'm just haven't. Yeah.
So, according to the screener, you prescribe Adenovate. Well, primarily you prescribe Adenovate, Eloctate, and Novoate. What do you remember about those products?
Yeah, I remember all those. Yeah. So we focus mainly on the hemophilia A patients, right?
Yeah.
Yeah. So we have been using Adenovate and Adenovate and Eloctate, Hydroxate, Novo 7, Novo 8, and Sensar. So including standard half-life and extended half-life agents. Most of those we have been used.
Okay. And what do you remember about these agents off the top of your head?
In regard to
anything.
Oh, anything? Yeah. So anything I remember is different half-life. Regarding the efficacy, safety profile, convenience use, expenses and insurance coverage, and the overall values. So among each of them, so certainly we have a different experience, and a different physician have a different experience. And I think that so far I will see there are several unmet clinical needs, and we're expecting more things going on. How we're going to evaluate clinical values for each of them in comparison to the current new agents, clinical trials, and gene therapy. So there's active discussions for each of them. Okay. So my general impression is some of the recombinant agents for hemophilia A going to be out of the market. So that's one impression. And the second, there are more active trial going on that is with new agents that is not only targeting factor 8 or 9 itself, but targeting on the regulatory loop components. With all those emerging new agents, including gene therapy, going to come to the market in the next few years, I think that will be some interesting changes going on that for the hemophilia field. And the third thing I'm feeling is about inhibitors. Now, those inhibitor positive patients, that is a major interesting thing that we are facing in the clinical practice. So for those patients, do we have any good options of the drug selections? So far, we have Hembra. We have other relatively ineffective agents that can be as alternative. But in general, I think this is the area that with a major unmet clinical need that we need to work on. Finally, I would think no matter which reagent we think about for the patients, the half-life is critical. So how we can maintain patients in compliance, that would be a key major things. Yeah, excuse me for 1 minute. Good, I'm back. Sorry. Yeah, go ahead.
Super. So what I'm going to do now is I'm going to share my screen with you.
Mm-hmm.
So you should now hopefully see something in front of you that says Product A Profile.
Yeah.
So please have a read of this and let me know when you're finished.
Okay. Prophylaxis, medium dosing, 3 days, medium ABR, patient is zero breathing. Frequency infusion, children, ABR, efficacy inhibitors. Oh. Mm-hmm. Yeah.
Okay. So what I'm going to do now is I'm going to show you 3 potential claims to support the development of a new communication campaign for this product.
Yes.
And I would like you to help me build what we're going to call a compelling and memorable story, which could differentiate Product A from competitor brands. And when I say compelling, I mean a story that builds a strong, attractive, and credible argument to encourage prescription of this product.
Okay.
On the next page, you should see in front of you 3 main claims: Claim 1, Claim 2, and Claim 3. So have a good read of these and let me know when you're finished.
So you mean I need to choose one from these 3?
Well, actually, I was going to ask you that as well, to rank the 3 in order. So rank them from most compelling down to least compelling. But obviously, I want to give you a minute to have a read of it first.
Okay. Yeah.
Okay. So tell me, how would you rank these 3 in order from most compelling down to the least compelling?
Yeah. I think each of these 3 claims, I feel all very good because they mainly explain different benefits or merits on this A. So I will rank claim 2 as the first one. Then I will, claim 3 will be second. Claim 1 will be a third paragraph.
Okay. So let's start with Claim 2 then. So tell me, what makes Claim 2 compelling?
Because you introduce what is origin, right? This, what is A? What I mean is it's a recombinant, right? As how they're coming from. And the purpose of trying to using A is because with low immunogenicity, that they will be effectively maintaining factor A function, but also reduce the possibility to developing inhibitors, right? So those claim, you're first giving introduction. So claim 2 is really play an important role on this aspect.
Okay. And the benefits you mentioned there, the immunogenicity, and restoring the factor VIII function, how are those benefits, would you say, how are they different from other brands you prescribe?
Yeah, so this very good point, because this seems to be the major benefit for A, right? So you have to show us the data to prove this is the case. So in comparison to other, this require back-to-back comparison. So we do not have solid data, even with all the available agents now so far we're using for A and B patients. We only based on what clinical.
Assuming that was true.
Yeah.
And assuming that what it claims to do in Claim 2 is true.
Yeah.
Is that different from other brands in your view?
We don't know. Because based on this description, we don't know. But I think we have to trust it, right? We have to trust it that this would depend on how this A was produced, the recombinant factor A was produced. Is that using whole molecule or only the major functional domain? To make a recombinant factor A constructions. Second is what kind of human cell line using? What is the kind of a post-translational modification has been occurred on these recombinant molecules, right? So